Welcome to the Deep Dive, where we extract the most important nuggets of knowledge to help you become truly well-informed. Today we're diving into something fundamental. These constant hidden battles happening inside every animal, including you. I mean, think about it. Your body is under relentless attack, not just viruses and bacteria, but sometimes even your own cells can go rogue. How does anything survive that?
It's an extraordinary feat of biology, really. And our mission today is to explore animal physiology, defense systems. And we're using animal physiology from genes to organisms. Second edition is our guide to unpack how animals, from the simplest sponges right up to us, protect themselves. You'll discover some truly ingenious strategies, from ancient always-on systems to highly sophisticated learned immunity. Expect some surprising facts, key experiments, and those crucial links between genes and function.
Okay, so let's start right at the foundation. It seems obvious, but maybe it isn't stressed enough. No animal could survive infancy without these defenses, right? Maintaining that stable internal environment, homeostasis, it's just impossible if your cells are constantly being injured or, worse, replaced by abnormal ones like cancer cells.
Exactly. It's absolutely foundational. And this isn't new. Organisms have been attacking each other for, well, billions of years. It's created this ongoing evolutionary arms race. One side develops a defense, the attacker evolves a way around it, which forces the first one to adapt again. It's this relentless pressure over huge timescales that shape the complexity we see.
It's amazing how critical it is. So critical, I gather, it can even influence mate choice in some animals, like those zebra finches.
Oh, absolutely. It's a fantastic example. Male zebra finches get these bright red and yellow pigments, carotenoids, from their plant-based diet. Makes their beaks look great to females. But here's the kicker, as you said. Those same pigments are also powerful antioxidants. They directly boost the bird's immune system.
So it's a genuine signal of health.
Precisely. Experiments showed males given extra carotenoids weren't just brighter, they had faster immune responses. And yes, females strongly prefer them. It's basically advertising. I'm healthy, I can fight off disease, our offspring will be strong.
But nature loves a trade-off, doesn't it? It's never quite that simple.
You've got it. There's often a cost. For instance, a study on male crickets found a link between stronger immune systems and sometimes, well, weaker sperm. Shows that mounting a powerful defense takes energy, right? Energy that might otherwise go into reproduction or other functions. It's a balancing act.
So when we talk about these internal defenses, immunity, we often split it into two main types, innate and acquired. What's the core difference there?
Okay, so innate immunity is your immediate, sort of ready-to-go defense. It's rapid, doesn't need prior exposure to a threat, and it acts against almost anything foreign. It's incredibly ancient. You find versions of it in all animals, even sponges, using cells that engulf invaders. People call it non-specific, but it does recognize specific common features on pathogens.
Okay, immediate and broad. Then acquired immunity. That's the one that learns.
That's the one. Acquired or adaptive immunity is all about learned responses. Highly specific. It targets particular invaders after you've encountered them once. And crucially, it remembers. It's slower to kick in the first time, maybe days or weeks, but it's incredibly precise. Antibodies are a classic example. We used to think only jawed vertebrates, fish, mammals, birds had this. But we'll touch on some newer findings later, suggesting other animals have something similar.
Got it. And just to clarify, for this deep dive, we're focused purely on these internal defenses, this immunity, not external things like, say, porcupine quills or toxins.
Exactly. We're looking inside the body. And this internal immune system, it basically has three main jobs. One, fight off invading pathogens, bacteria, viruses, parasites like the tick that gives you Lyme disease. Two, cleanup crew, removing worn-out cells, tissue debris, helping with repair. And three, immune surveillance, constantly checking for and destroying abnormal or mutant cells. That's your internal defense against cancer.
Which leads to a really fundamental question. How does the system even know what's self and what's non-self? That seems absolutely critical.
It is the critical distinction. There's a great visual for this ancient ability, the anemone clone wars. You see these sea anemones growing in clusters. Well, each cluster is often a clone, genetically identical. If two different clones start to grow near each other, they recognize the other as non-self, based on unique molecules on their surfaces, and they literally fight. Using stinging tentacles, creates these empty zones between them. It's a very visible example of self versus non-self recognition, quite similar to what happens inside us.
Wow. So, okay, down at the molecular level inside us, how does that recognition work? What's doing the seeing?
A lot of it comes down to pattern recognition receptors, PRRs. Think of them as the eyes of the innate immune system. A famous example is the toll receptor in fruit flies. When that gene was messed up, flies got overrun by fungi. Then, similar receptors, the toll-like receptors, or TLRs, were found in mammals. They sit on our immune cells and recognize common foreign patterns like lipopolysaccharides, LPS on bacterial walls, or specific types of viral RNA, things that scream invader.
And once they spot something, how do the immune cells talk to each other, coordinate the defense?
Two main ways. They release signaling molecules called cytokines. You can think of them as immunohormones. They can act locally on neighbors or travel through the blood like hormones for a wider response. Interleukins are a big group of these invertebrates. The other way is direct contact. Cells can literally touch, form a temporary junction called an immunosynapse, and pass signals directly that way.
You know, it's interesting. We don't just rely on our own defenses. Sometimes animals, including us, sort of borrow defenses from other organisms.
Oh, definitely. Humans have been doing it for ages. Aspirin. Originally from willow bark, it's a plant defense compound we repurposed. And you see it elsewhere. Chimps swallow specific leaves, Aspilia leaves, whole, to get anti-parasitic compounds. Capuchin monkeys rub millipedes on themselves. The millipedes produce benzoquinones that repel mosquitoes.
Birds lining nests with certain plants too, right? To fight parasites.
Exactly. It's nature's pharmacy in a way. Life is incredibly resourceful at finding ways to protect itself.
That's pretty amazing. Okay, let's shift focus to our innate immunity again, specifically the first and second lines of defense, thinking mainly about mammals. First line, barrier tissues.
Right, the walls. And the skin is the big one, our largest organ. It's a formidable mechanical barrier. Layers of tough, keratinized cells tightly linked, constantly shedding and being replaced. Largely waterproof, keeps most things out.
But it's not just passive, is it? It fights back too.
Absolutely. It's an active barrier. Sweat glands secrete antimicrobial peptides, like dermcidin, that help protect wounds. Sebaceous glands produce sebum, that oily stuff. Waterproofs, yes, but also inhibits microbe growth. Plus, you have specialized cells within the skin itself, like keratinocytes, that produce other antimicrobial compounds and signal other immune cells if there's a breach.
And it's not just skin. Other openings have barriers too.
Oh yeah. The digestive tract is a major battleground. Think about what you eat. So you've got defenses like lysozyme and saliva that kills bacteria, powerful stomach acid, specialized immune tissue, GLT lining the gut, producing its own antimicrobials, genitourinary system, acidic urine, vaginal secretions, sticky mucus traps, respiratory system, hairs in your nose, tonsils, adenoids, and that amazing mucus escalator.
The cilia beating upwards.
Exactly. Constantly sweeping mucus with trapped debris up and out, plus macrophages waiting in the lungs. All crucial first lines.
Okay, so if something does breach those barriers, that's when we get inflammation, right? The redness, swelling, heat, pain. What's the point of that? Seems counterproductive sometimes.
It feels bad, but it's a highly coordinated, essential emergency response. The main goals are to stop the damage spreading and to quickly flood the area with defensive fluids, immune cells, and proteins. So, say bacteria get through a cut. Resident immune cells, like macrophages, are already there. They recognize the invaders using those TLRs, or they sense signals from your own damaged cells and they kick things off.
What's the first step that causes the noticeable signs?
Almost immediately, tissue cells called mast cells detect the injury or invasion and release histamine. Histamine is a key alarm signal. It makes local small blood vessels dilate, widen, increasing blood flow. That causes the redness and heat. And crucially, it makes the vessel walls leaky, more permeable. Fluid, proteins, and cells escape into the tissue. That's the swelling, which can press on nerves causing pain, basically setting up an emergency zone.
Right. And how do more defenders get to that zone?
Chemical signals called chemotaxins are released, acting like beacons. They draw immune cells, leukocytes, out of the bloodstream. Neutrophils are usually the first responders. Highly phagocytic, meaning they eat invaders. They squeeze through the leaky capillary walls. Then monocytes arrive and mature into more macrophages, also big eaters. They engulf bacteria, debris, damaged cells. They also release toxic chemicals to kill pathogens directly. Meanwhile, clotting factors leak out and form fibrin clots, kind of walling off the area to hopefully contain the infection. Then eventually, cleanup and repair begins.
This local fight can sometimes trigger a whole body response too, like fever. Why do we get hot?
Yeah, those activated macrophages release certain cytokines like IL-1 that travel to the brain, specifically the hypothalamus, your body's thermostat. They act as endogenous pyrogens, telling the hypothalamus to crank up the set point. Moderate fever seems beneficial. It can enhance phagocyte activity, speed up enzyme reactions, and maybe inhibit some bacteria by messing with their iron uptake.
Even lizards do it, behaviorally.
Yeah, sick lizards will actively seek out warmer spots. Suggests it's a very old conserved defense strategy. Though very high fevers are dangerous, of course.
Okay, another piece of the innate puzzle, the complement system. Sounds complex.
It's a group of about 30 plasma proteins,
Yeah.
are normally inactive, just circulating. But when activated, they cascade, one activating the next. The end result is often the formation of a membrane attack complex, or MAC. This MAC literally punches holes in the membranes of microorganisms, causing them to burst open, like those ancient antimicrobial peptides, but maybe more targeted.
And how does it get activated?
Two main ways. One is innate, the alternative pathway, where complement proteins directly bind to certain carbohydrate chains common on microbes. The other way, the classical pathway, involves antibodies, part of the acquired immune system, binding to the microbe first, and then complement binds to the antibodies. Shows how the systems intertwine. Complement proteins also help amplify inflammation.
Got it. And for really rapid responses, especially against viruses and cancer cells.
You have natural killer cells, NK cells. These are lymphocyte-like cells, but they act non-specifically. They can recognize and kill virus-infected cells or tumor cells on first exposure without prior sensitization. They lease the target cell membranes, acting as a really important early defense before the more specific T cells get fully activated. And then there's interferon. When a cell gets infected by a virus, it releases interferon.
The whistleblower.
Exactly. Interferon doesn't help the infected cell much, but it signals nearby healthy cells. They respond by producing enzymes that interfere with viral replication, blocking viral mRNA, inhibiting protein synthesis. It's a warning system. Plus, interferon has anti-cancer effects too.
So understanding all this helps explain how things like anti-inflammatory drugs work, doesn't it? NSAIDs, steroids.
Precisely. NSAIDs like aspirin and ibuprofen block some inflammatory pathways. Glucocorticoids, like prescription cortisol, are powerful suppressors of inflammation and overall immune responses. And your body's own cortisol, released during stress, normally acts as a brake, a negative feedback, preventing the immune system from overshooting and causing too much collateral damage. It's a crucial balance.
Okay, let's move on to the other major branch, acquired immunity. The master orchestrator, the third line of defense. This is where things get really specific and sophisticated, especially in us jawed vertebrates.
Absolutely. This is the system that provides those highly selective attacks, tailored to specific invaders, and importantly, remembers them after the first encounter. Its roots, in terms of our understanding, go way back to Edward Jenner in 1796, noticing cowpox protected against smallpox. That's the birth of vaccination, harnessing this memory. It's a complex system, likely costly to run, but the benefit is immense. The ability to generate potentially millions of different receptors from relatively few genes, allowing adaptation to new and evolving pathogens. Hugely important for long-lived animals.
And it has two main arms you mentioned.
That's right. First, antibody-mediated immunity, sometimes called humoral immunity, because it involves substances in the body fluids or humors. This relies on B lymphocytes, B cells. They transform into plasma cells that secrete antibodies, which target extracellular threats like bacteria or free viruses.
Okay, B cells and antibodies, the second arm.
Cell-mediated immunity. This involves T lymphocytes, T cells, particularly cytotoxic T cells, which directly attack and destroy the body's own cells that have become infected or cancerous, dealing with intracellular threats.
And you also mentioned helper T cells and regulatory T cells playing key roles.
Yes, they're crucial coordinators. Helper T cells are like the generals. They secrete signals, cytokines, that turn on and amplify the activity of both B cells and cytotoxic T cells. They're central. Regulatory T cells, or TREGs, do the opposite. They put the brakes on, suppressing immune responses to prevent overreaction or autoimmune attacks. It's all about balance.
And the devastation of HIV makes sense then because it targets those helper T cells.
Exactly. It takes out the coordinator, crippling the whole acquired immune response.
Where did these B and T cells come from and hang out?
They originate from stem cells in the bone marrow, like all blood cells. B cells mature right there in the bone marrow, B for bone marrow in mammals, or bursa of Fabricius in birds where they were first discovered. T cells, however, migrate to the thymus gland, hence T cells. The thymus is like a school where they undergo crucial maturation and education to learn self from non-self. Then mature lymphocytes circulate and also reside in specialized lymphoid tissues, lymph nodes, spleen, tonsils, patches in the gut, and skin, salt, strategically placed to encounter invaders.
And what exactly are they responding to? What triggers them?
They respond to antigens. An antigen is typically a large, complex molecule, usually a protein or large polysaccharide, that is recognized as foreign and triggers a specific immune response against itself.
Right. Let's zoom in on the B cells first, the antibody factories.
Okay, so each B cell has unique receptors on its surface, B cell receptors or BCRs. These are basically membrane-bound versions of the antibody that the cell will eventually secrete. Each BCR is exquisitely specific for one particular antigen. When that antigen binds to the BCR, it activates the B cell.
What happens when it's activated?
It starts dividing rapidly and differentiates. Most become plasma cells. These cells are incredible protein factories. Their internal machinery, the endoplasmic reticulum, swells up as they churn out thousands of antibody molecules per second. They secrete these antibodies into the blood and lymph, but they're short-lived, maybe 5-7 days. A smaller number become long-lived memory B cells, which we'll come back to.
And the antibodies themselves, these immunoglobulins or Ig, what do they look like and do?
They're typically Y-shaped proteins. The two arms of the Y contain the antigen-binding sites, the Fab regions. These are highly variable, providing the specificity like a lock for the antigen key. The tail of the Y, the FTC region, is more constant within a class and determines the antibody's function, what it does after binding. There are different classes, like IgM, often the first type made, also acts at the BCR. IgG, most abundant main player against bacterioviruses, crosses placenta. IgE, fights parasites, involved in allergies. IgA, found in secretions like mucus, tears, milk, and IgD, mostly on B cell surfaces, function less clear.
You mentioned earlier they don't usually kill directly, so how do antibodies work?
Right, they mainly hinder invaders or amplify other defenses. They can physically hinder by neutralization, blocking a virus or toxin from binding to a host cell, or by agglutination, clumping invaders together, making them easier to clear. But their biggest role is amplifying innate defenses.
How so?
They activate the complement system via that classical pathway we mentioned, leading to the MAC punching holes. They act as opsonins, coating invaders to make them much easier for phagocytes like macrophages to grab and eat. IgG is a great opsonin. And they can also flag targets for destruction by NK cells.
Okay, this specificity is amazing. How does the body generate B cells ready for almost any conceivable antigen before it even encounters it? That seems impossible.
It does seem impossible. The answer is the clonal selection theory, a Nobel Prize-winning idea. Basically, during development, your body generates a massive diversity of D cell clones randomly. Each clone is pre-programmed with a unique BCR, ready for a specific antigen it might never even meet. When an antigen eventually enters the body, it effectively selects the B cell clone that happen to have the matching BCR. Only those selected clones are then activated to multiply.
So the antigen picks the winner rather than inducing the B cell to make the right antibody.
Exactly. The diversity is pre-existing. That initial activation leads to the primary response. Those chosen B cells proliferate, make plasma cells, secrete antibody. This takes time, maybe a week or two to peak, but crucially, some of those activated B cells become long-lived memory cells. They don't secrete antibodies right away, they just wait.
And that's the key to long-term immunity and vaccines.
Precisely. If the same antigen enters the body again, months or years later, those memory cells are already there, primed and ready. They launch a secondary response that is much faster, much stronger, and lasts longer than the primary response. Often, you eliminate the infection before you even feel sick. Vaccination works by deliberately inducing that primary response and generating memory cells using a safe, non-disease-causing version of the antigen.
But how do we get that initial massive diversity, millions of different antibodies from a limited number of genes?
Through a remarkable process of genetic recombination. During B cell development, different segments of the antibody genes bits called V, D, J, and C sections are literally cut, shuffled, and spliced together in a huge number of random combinations. It's like having a few Lego bricks, but being able to build millions of different structures. Add in some sloppiness and then splicing and even some later mutations in activated B cells, and you get immense diversity.
Like evolution within the immune system itself.
Sort of, yeah. Random variation arises, and the antigen selects the fittest binders, which then proliferate. It's quite Darwinian in principle.
Okay, so that's active immunity. Your body makes its own response. What about passive immunity again?
Passive immunity is when you receive pre-made antibodies from another source. The classic example is a mother passing antibodies to her baby, either across the placenta before birth, IgG, or in breast milk, especially colostrum, IgA. It provides crucial temporary protection while the baby's own immune system is still developing. But it's passive. The baby isn't making the antibodies, so it wears off after weeks or months. You don't get memory cells this way.
Right. Now let's switch to the T lymphocytes, the cell-mediated arm, the covert operators, you called them.
Yeah, because unlike B cells releasing antibodies into the fluids, T cells generally have to make direct contact with their targets. They specialize in dealing with threats inside our own cells, like viruses replicating within or cancer's changes.
They're also antigen-specific, like B cells.
Yes, highly specific, and they also undergo clonal selection and form memory cells, giving us long-term cellular immunity. Each T cell has a unique T cell receptor, or TCR, but here's a key difference. T cells don't recognize free-floating antigens. They only recognize antigen fragments that are presented to them on the surface of another body cell, displayed by special molecules called MHC molecules. That's part of their thymic education.
MHC, major histocompatibility complex. That sounds important.
Crucial. MHC molecules are like billboards on the cell surface. There are two main types. Class I MHC is found on almost all nucleated body cells. It displays fragments of proteins from inside that cell. If the cell is infected with a virus, it will display viral fragments on its Class I MHC. Class II MHC is found only on specialized antigen-presenting cells, APCs, like macrophages, dendritic cells, and B cells. They display fragments of things they've engulfed from outside.
Okay, so T cells need this MHC presentation. You mentioned 3 types of T cells.
Right. First, cytotoxic T cells, or CD8 plus T cells. These are the hitmen. They recognize foreign antigens, like viral proteins, presented on Class I MHC molecules on infected body cells. They bind to the infected cell via their TCR and MHC, form an immunosynapse, and then kill it, often by releasing proteins called perforins that punch holes, or granzymes that trigger apoptosis program cell death. This stops the cell from being a virus factory.
Effective. What about helper T cells, CD4 plus T cells?
These are the master coordinators, the generals we mentioned. They don't kill directly. They recognize antigens presented on Class II MHC molecules on APCs. When activated, they secrete cytokines that are essential for activating and amplifying both B cell responses, leading to antibody production, and cytotoxic T cell responses. They also boost macrophage activity, absolutely central to most adaptive immune responses.
Hence the HIV problem. And the third type, regulatory T cells.
TREGs, often CD4 plus and CD25 plus. These are the suppressors. They act as a crucial check, dampening immune responses to prevent them from going overboard, minimizing damage to healthy tissue, and helping maintain tolerance to self. There's a lot of interest in manipulating TREGs for treating autoimmune diseases or preventing transplant rejection.
You mentioned antigen-presenting cells, APCs, are needed to show antigens to T cells. Tell us more about them.
Right, T cells need that formal introduction. The main professional APCs are macrophages, B cells, which present antigens they bind via their BCRs, and especially dendritic cells. Dendritic cells are amazing sentinels. They reside in tissues, especially barriers like skin, where they're called Langerhans cells. They continuously sample their environment. If they engulf an invader or detect danger signals from damaged cells, like uric acid, they get activated, process the antigen, load it onto their MHC molecules, and then travel to the nearest lymph node to present it to T cells, initiating the adaptive response.
So these MHC molecules are the key platform for presentation. Are they the same in everyone?
No, far from it. MHC genes are incredibly variable in the population. Hundreds of versions exist, though each person only inherits a few. This means your set of MHC molecules is almost unique, like a biochemical fingerprint. This diversity is crucial for the population's ability to respond to a wide range of pathogens. But it's also why finding a match for organ transplants is so difficult. The recipient's T cells will recognize the donor's different MHC molecules as foreign.
And this relates back to immune tolerance preventing attacks on self.
Absolutely. The immune system has multiple checks to avoid attacking tissues bearing your own MHC molecules with normal self-peptides. During T cell development in the thymus, cells whose TCRs bind too strongly to self-MHC self-peptide complexes are usually eliminated. That's clonal deletion. There's also clonal energy, where self-reactive cells that escape deletion are rendered unresponsive, and receptor editing in B cells, where they can quickly change their BCR if it's self-reactive, plus ongoing suppression by those TREGs.
But sometimes it fails.
Yes, that leads to autoimmunity diseases like type 1 diabetes, rheumatoid arthritis, multiple sclerosis, where tolerance breaks down and the immune system attacks self-tissues. The exact triggers are complex and not fully understood.
What about cancer? Does the immune system fight that too?
Yes, definitely. The concept is called immune surveillance. Cancer cells arise from normal cells through mutations. These mutations can create new, abnormal proteins that T cells can recognize as foreign when displayed on the cancer cell's MHC class I molecules. So, cytotoxic T cells and K cells and macrophages are constantly patrolling and, ideally, eliminating newly formed cancer cells before they can grow into tumors. Interferon also plays a role.
But cancer sometimes wins that battle.
Unfortunately, yes. Some cancer cells develop ways to evade detection, maybe by reducing their MHC expression or by producing signals that suppress T cells or even recruiting Tregs to protect them. It's another evolutionary arms race happening inside the body.
It's becoming clear how interconnected everything is. The immune system isn't just floating around on its own.
Not at all. There's a deep, intricate connection between the immune system, the nervous system, and the endocrine hormone system. It's often called the neuroendocrine immune loop. For example, immune cytokines like IL-1 can signal the brain to induce fever, but also to activate the stress response, leading to cortisol release. Cortisol has metabolic roles, but it also suppresses immune function, a feedback loop. Nerves directly innervate immune organs. Immune cells have receptors for neurotransmitters and hormones. Acute stress might briefly boost immunity via adrenaline, but chronic stress, often via cortisol, generally suppresses it. This provides a biological link between your psychological state and your susceptibility to illness.
Amazing complexity. Let's zoom out now to some broader evolutionary questions. How did this incredibly complex acquired immunity of jawed vertebrates even come about? You hinted at a viral connection.
It's a fascinating hypothesis, yeah. It might have arisen relatively suddenly due to an evolutionary accident. The key seems to be genes called RAG1 and RED. These genes code for an enzyme, a transposase, that performs the random cutting and pasting of gene segments needed to create the vast diversity of B-cell receptors and T-cell receptors.
The gene shuffling.
Exactly. Now, RAG genes are found in all jawed vertebrates, sharks, fish, us, but not in jawless fish, like lampreys or hagfish. And transposases are often found in viruses and jumping genes. So the theory is that maybe 450 million years ago, a transposon, possibly carried by a virus, inserted itself into the genome of an ancestral jawed fish. And instead of just wrecking things, its cutting-pasting ability got co-opted to shuffle immune receptor genes. We might owe our adaptive immunity to a lucky viral infection in an ancient fish.
That is truly wild, a profound accident of evolution. What about defense systems in other animals, then?
Well, we're learning more all the time. Sharks, for example, are ancient jawed vertebrates. They have B and T cells, but their system differs from ours. They seem to rely less on the random mutation part for diversity and have many more preformed antibody gene clusters, perhaps for faster responses to common marine pathogens. Shows alternative successful strategies. Lampreys, the jawless fish, independently evolved their own form of adaptive immunity using completely different molecules called variable lymphocyte receptors, VLRs, generated by shuffling different gene segments. Convergent evolution in action.
And invertebrates, any signs of learning or memory there?
This is a really hot area. While they lack the vertebrate B cell system, we are seeing hints of adaptive-like responses. For example, water fleas, Daphnia, exposed to a bacterium produce offspring that are more resistant specifically to that bacterium. Fruit flies, given a small dose of bacteria, survive a later lethal dose better. And arthropods have this incredible molecule called DSCAM. Through alternative splicing of its gene, they can generate tens of thousands of variants. These act as immune receptors on their hemocytes, blood cells, and can even be secreted, potentially acting like antibodies to enhance phagocytosis. Experiments show boosting specific DSCAM variants in mosquitoes help them fight off later infections.
So it's maybe not the same lifelong specific memory as ours, but something more sophisticated than just pure innate immunity.
Exactly. It blurs the lines a bit. Whether these invertebrate systems truly exhibit lifelong memory in the same way is still debated and under intense research, but it shows the innate acquired distinction might be less absolute than we once thought, and that diverse solutions to pathogen defense have evolved.
So wrapping this up for you, our listener, we've journeyed through these layers of defense, from molecules recognizing non-self right up to evolutionary arms races spanning millions of years. It's just an incredible, multi-layered, interconnected system.
Absolutely. And understanding these defense systems, even at this level, gives you a really profound insight into the very basis of health and survival. It's in the machinery that allows complex life to persist in a world full of threats.
As you go about your day, maybe just take a moment to appreciate this constant, silent battle happening inside you. And consider this. How might these ancient defense mechanisms, shaped by past evolutionary pressures, cope with the new challenges of our modern, rapidly changing world, new pathogens, environmental toxins, maybe even novel therapies? It's an ongoing story.
It really is. We sincerely thank you for joining us for this deep dive into animal defense systems, and thank you for being part of our last-minute lecture family.